So let's first look at the three major classes or paradigms in anti-cancer therapeutics. And here we're really looking at pharmacological agents. So we're not talking about surgery or radiation. So there are three major paradigms. So the first one, and probably one of the oldest and still most commonly used, is the cytotoxic chemotherapy. You probably heard that term as just referred to as just chemotherapy. And these are drugs that non-specifically hit all rapidly proliferating cells, whether cancer or normal. And it's still the mainstay and the core of clinical cancer therapy. Then some of the newer agents are the targeted therapies, which generally are designed to only affect cancer-specific molecular alterations and therefore tend to be less toxic and more specific to cancers. They're usually informed by the individual genetic alterations that are found in cancers or upregulation of specific pathways. And then the third and now really booming area is the immunotherapy, which takes advantage of the body's own defenses to track down and fight cancer cells. So immunotherapy, broadly, it's just the treatment of any disease, not necessarily cancer, by inducing, enhancing, or in some cases suppressing an immune response. And specifically in cancer treatment, immunotherapy is supposed to stimulate the immune system to track down and attack tumor cells through similar mechanisms that it attacks bacteria and viruses. And also, cancer immunotherapy can supplement and enhance the immune system with exogenously provided man-made components. So the objectives of this lecture are, after the end of this lecture, I'm hoping you would be able to understand the basic principles of anti-tumor immune response and a very bird's eye view of the anti-tumor response. You would know the basic mechanisms of action for each class of cancer immunotherapy, and you would be able to understand the challenges and limitations of immunotherapeutic approaches, including some specific toxicities that are associated with these treatments. So I just mentioned that cancer chemotherapy is one of the newer and booming approaches, although you may or may not realize that actually it's been around for quite a long time, since the end of the 19th century, actually. And William Coley, who is known to be the father of cancer chemotherapy, introduced so-called Coley's toxin, which was a mixture of heat and activated bacteria, which were injected intratumorally. And so he observed some really stunning results with tumor regression, and for a while it was very highly advertised as the new groundbreaking cancer therapy. However, he had some issues with reproducibility of his protocols. He didn't really maintain a good lab notebook, so to say, and therefore, after a while, there was a lot of skepticism about this treatment, and so he fell out of favor, and the cancer immunotherapies were not resurrected until much later. However, at that time, Dr. Coley, who worked at the Memorial Sloan Kettering Cancer Center in New York City, was encouraged to explore another very commonly used approach to cancer treatment nowadays, to radiotherapy. And so he also did a lot of work to advance that into the clinic with great success. So let's look, again, as a very superficial bird's eye view at the cancer immunity cycle. And so you can see that it usually starts at the tumor site, because a lot of tumor cells, for different reasons, die. They either undergo apoptosis or they die through necrosis, and that leads to release of a large load of cancer antigens. So also cancer cells, still even live ones, present a lot of their antigens on their surface. And so these tumor antigens are being picked up by the dendritic cells for processing and activation. And so the next step really happens in the lymph node, where these dendritic cells can now prime and can present these antigens to T cells and prime and activate the T cell response. And so now activated T cells start patrolling the body. And so once they extravasate, find the tumor and extravasate, they're able to recognize the tumor antigens on the surface of tumor cells and mediate tumor cell killing. And that eventually restarts the cycle, because after the tumor cell has been killed, there's again an even larger release of cancer antigens to start the cycle again. And so again, this cross-presentation of tumor antigens, let's look at it a little bit more closely. So as I mentioned, some tumor antigens are being, a lot of them are being presented on MHC class 1 on tumor cells that are being taken up, captured by the host antigen-presenting cells, which take them up and process them for presentation again on the surface of APCs that now present them to CD8 cytotoxic T cells that become primed and activated, and now can patrol the body in search of the tumor cells for degradation. So there are several types of tumor antigens. So first of all, we all know that normal cells display their normal self-antigens, and that is associated with with no T cell response, as these are normal self proteins. And then there are several types of tumor antigens that can be presented. These are either mutated self proteins, although because a lot of tumors have a really increased rate of mutagenesis and genetic instability, that leads to the creation of slightly broken or slightly different variants of self-proteins that are now which may or may not contribute to tumorigenesis. A lot of them are just bystanders. However, these are now unique antigens to the tumor that are differ from self-antigens, and that can be used as to prime anti-tumor immune response. These may also be products of oncogenes or mutated tumor suppressor genes. So in this case, these are the proteins that do directly participate in the cancerogenesis process. And these can also serve as tumor-specific antigens for the cytotoxic CD8 positive T cell response. Those can be just overexpressed or aberrantly expressed self-proteins. And so here are some examples. And another interesting example is, for example, breast cancers in postmenopausal women may oftentimes express milk proteins that are not normally expressed at that age, and potentially can be also harnessed as aberrantly expressed self-antigens for tumor targeting. And finally, as we're now finding that actually a large proportion of tumors are either being driven by oncogenic viruses or just display abnormal reactivation of endogenous or exogenous viruses. And so these viral antigens can also serve as tumor-specific antigens to drive the immune response. So let's first look at the bone marrow transplant, which you may not have realized that that's actually one of the earliest forms of cancer immunotherapy. So in this case, bone marrow transplant is mostly used to treat patients with leukemia and other hematologic malignancies. And so after initial response to cytotoxic chemotherapy, the patient's diseased bone marrow is being wiped out by intensive chemotherapy and priming regimens. And after that, lethal damage to the bone marrow is being rescued by providing a donor bone marrow, which not only reconstitutes hematopoiesis or blood production, but also it serves a very important function of graft versus tumor effect, which means that the donor-derived normal bone marrow will provide immune response, residual immune response that would track down and kill potential residual persistent tumor cells. And that, in large part, is the mechanism of success of bone marrow transplant, because the perfect antigen matching is almost never possible. However, this presence of the slightly unmatched donor bone marrow also presents a significant challenge, so-called graft versus host disease, where the donor immune system may also recognize the normal self-tissues as foreign and attack them. And unfortunately, extensive graft versus host disease is very hard to manage clinically and may sometimes be even deadly. But we're gonna talk about some other more specific types of cancer immunotherapy. We're gonna cover different types of monoclonal antibodies. We're gonna look very little at immunomodulators, and then we're gonna talk about some newer developments in that area, such as cancer vaccines, checkpoint inhibitors, and finally the chimeric antigen receptor T cells. So let's start with monoclonal antibodies. First, let's look at some nomenclature. So initially, recombinant antibodies were mostly produced in mice, and therefore they have their constant and variable regions, all consisting of having mouse structure. And these antibodies can be produced from hybridoma in large amounts and can be infused into humans and have a very substantial effect. However, due to their pretty much exclusively murine structure, they are being recognized as foreign and attacked by the endogenous human immune system, and therefore may produce immune-associated toxicities, such as pretty much just out of proportion inflammatory response with so-called cytokine storm, and also decreases the stability and efficiency of the antibody. And to overcome this, there are different strategies how to replace larger and larger proportions of this antibody with human regions. And so pretty much it can be a chimeric structure where 75% of the antibody is human, and only largely the variable regions are human, and we can tell which type that is by the suffix, and so the chimeric would be the Z. And it can be further humanized, where 95% of the structure is now being human, with 5% coming from the mouse, which would have the suffix ZU. Then there can be a mixed chimeric or humanized structure, and we're just mixing the chimeric for Z and the ZU for humanized, getting us the ZZU. And finally, nowadays we can also derive a fully human antibodies that would have the suffix U. And here, obviously, the ending MAB is for monoclonal antibodies. And so just knowing how to read the nomenclature, you would immediately know the structure of the antibody. So, for example, when you see rituximab, you can immediately tell that it's a chimeric antibody. So antibodies, we would use several examples here. So the most probably one of the most commonly used antibodies for immunotherapy is the rituximab. It's the anti-CD20 chimeric monoclonal antibody, and CD20 is a B cell surface marker. It's a B cell specific antigen that is being expressed on all B cells, whether they're normal or malignant. And so CD20 right now is one of the core therapies for refractory B cell malignancies, such as non-Hodgkin's lymphoma and large cell B cell lymphomas. And also, it's also being used for some autoimmune diseases with a large B cell component. And so, however, since it's a anti-B cell antibody, so it pretty much affects all B cells, whether normal or abnormal. And it's also a chimeric antibody, which means that it still has a mouse murine portion, which really drives the adverse effects that are being seen during the use of rituximab. So the first dose might be associated with infusion-related symptoms, which are pretty mostly allergy-related symptoms due to the murine part of the antibody. And these symptoms sometimes can be severe and sometimes would require desensitization protocols, pretty much like desensitization for any other severe allergic reaction. But then the later side effects would actually be associated with an on-target toxicity associated with B cell depletion. It would either be immunodeficiency associated with depletion of normal B cells, and sometimes it's reactivation of endogenous viruses. And also, oftentimes it might lead to bowel perforations because of the depletion of B cells in the Peyer's patches, or when the malignancy infiltrates into the gastrointestinal tract. And so there's also sometimes the delayed onset neutropenia because those cells can, neutrophils have a high affinity Fc gamma receptors that bind to these antibodies and then can result in non-specific elimination of the cell coated with an exogenous antibody. So another example would be the trastuzumab, also known as Herceptin, which is anti-HER2 or ERBB2 humanized monoclonal antibody. However, this is less considered to be an immunotherapy and more considered to be a targeted therapy because the predominant mode of action is actually the inhibition of the oncogenic signaling pathway, which is driven by overexpression of ERBB2, which is different from the mechanism of action of rituximab, which is predominantly driven either by antibody-dependent cell-mediated cytotoxicity, where the rituximab antibody recognizes the B cell, and then this bound antibody is then being recognized by the effector cells that bind to the antibody through Fc gamma receptors and mediate the direct cell killing. Or also another mechanism of rituximab mechanism of action is the complement-dependent cytotoxicity, where again the antibody bound to the target cell would then recruit the complement, which then unleashes the formation of the membrane attack complex that leads to cell lysis. And so direct effects associated with the inhibition of the CD20 signaling, well, actually the downstream effects of which are not very well studied, are a minor component in this anti-B cell action of rituximab. And so there are different anti-CD20 antibodies that are being clinically used, and they all target slightly different parts of the CD20 surface antigen, and as a result can be either combined, or if resistance or hypersensitivity develops to one of the antibodies, then it can be switched to the use of a different antibody. So the next variation of using antibody as an anti-cancer treatment is the antibody drug conjugates. And here the idea is that we can use the antibody to specifically find and target the cancer cells, and then the cancer cells are not going to be killed by the antibody-mediated effects, but rather by delivering a payload. And payloads might be very different. Those might be cytotoxic agents, those might be radioisotopes, those even can be cytokines. And so here I have two examples for you. So either the ibrutinib deruxtecan, which is again an anti-CD20 monoclonal antibody, which is now conjugated to a radioactive isotope. And so as a result, this antibody specifically delivers the radioactivity to the B cells in the tumor and decreases the radioactive exposure to the rest of the body. And so that allows to concentrate the radioactivity in the tumor and minimize the side effects of radioactivity. And so again, it's an anti-CD20 antibody, so it's being used in relapsed refractory follicular B-cell non-Hodgkin's lymphoma and similar malignancies. Another example, and these are just some examples because there are multiple combinations of antibodies with payloads that are available, either already FDA approved or in development. So another example would be the gemtuzumab ozogamicin, also known as Mylotarg. This time, it's an anti-CD33 humanized monoclonal antibody conjugated to an anti-tumor antibiotic that, after internalization, pretty much leads to extensive DNA damage. So CD33 is expressed on surface of acute myeloid leukemia cells. And so in this case, the CD33 antibody conjugated to this DNA damaging agent allows the delivery of the DNA damaging agent specifically to the leukemia cells and not other cells. So in this case, CD33 tends to be expressed exclusively on leukemia cells and not on normal myeloid cells. And so this is a pretty old drug. It was initially FDA approved in 2000, but then because of the extensive pretty significant side effects and also some of the many cases of resistance was voluntarily withdrawn 2 years later. However, with more careful selection of appropriate patient population and with some other precautions over the appropriate dosing regimens, it was reapproved in 2017 and is an important tool in the arsenal of oncologists in managing acute myeloid leukemia, CD33 positive acute myeloid leukemia. And then now one of the newer developments in the antibody department is the bispecific T-cell engagers that are also known as BiTEs. And one of the most successful examples is blinatumomab, which was FDA approved a few years ago. And it is used to target the CD19 positive B cell tumors as a second line treatment. And so the idea behind BiTEs is that it has two components. So it pretty much has variable regions from two different antibodies that are just linked together by a flexible linker. And one of them is an antibody that targets the cancer cell. So in this case, it's an anti-CD19, and CD19 is another B cell specific antigen. However, now the second part is an anti-CD3 antibody, and CD3 is a receptor on the surface of T cell that is necessary for T cell activation. And so by having these two parts pretty much stapled together, the BiTEs can first bind to a tumor cell and then recruit the T cell directly for cancer cell killing, which would also skip the MHC, so major histocompatibility complex antigen presentation, and would also bypass the T cell receptor activation. So it's just direct recruitment of the T cell to the tumor cell to facilitate direct cell killing. So now let's look very briefly at immunomodulators, which are pretty much represented by different cytokines. And so we're covering them because these are some of the earliest FDA-approved immunotherapies, so immunomodulating therapies for cancer, but because of their generally very high toxicity, they now have very limited use. But the first one is a high-dose recombinant interleukin-2, which was FDA-approved in the 1990s. And again, this is pretty much the first effective modern immunotherapy for cancer, which is being used for metastatic renal cell carcinoma and also occasionally for metastatic melanoma. And just massive doses of interleukin-2 promote differentiation of T cells into effector T cells in the presence of the tumor antigens. However, there are multiple issues associated with this very non-specific therapy because they may promote T regulatory cells that may lead to some paradoxical responses, and also as a high-dose cytokine, it can lead to serious, potentially lethal immunoactivation effects. And similarly, the recombinant interferon alpha, which is approved for hairy cell leukemia and a couple of other malignancies. So the mechanism of action here, it induces expression of MHC class 1 on tumor cells, and that really promotes the presentation of tumor antigens on the surface of tumor cells and drives maturation of dendritic cells that can now prime and activate tumor-specific T cells. However, because it's an interferon alpha, and so interferons are pretty much the cytokines that are being released in massive amounts during viral infections and drive all the symptoms that are associated with viral infections, such as high fever and other symptoms. So it may be associated with significant organ toxicity, and therefore its use is pretty much being limited by this toxicity.